right, Josh, back over to you. It's hour 12, so we're
Yeah, I was sitting here chuckling with you, not knowing what time it was. So
yeah,
I'll keep it going.
All right. I'm going to see everybody else at 5 in the morning, but good luck to the rest of you tonight, and I'll be watching on my phone. All right, everybody.
Bye, Greg.
Bye.
All right. I will sit here and talk to myself for another minute. We are on hour 12. Just to orient you, if your name is Greg Gonzalez and you need to know what hour it is 10 o'clock Eastern, 8 o'clock p.m. Mountain Time, and 7 p.m. Pacific. I shouldn't skip Central, so that means it's 9 o'clock Central. I am delighted to host for the next hour, and we will start with a friend and colleague on a topic that is near and dear to my heart. If we can have Rachel Bender-Ignacio come onto the screen. Oh my God, there she is.
Hi, Josh.
So nice to see you. I feel like I should bring energy back up, but I know that actually, because it's like a little bit later, but I figure Rachel's here, so energy is surely going to be high. I will pass it to you, my friend, and I will shut my mouth.
Sounds good. Well, thank you for having me on. The sun is still up over here on the West Coast, so we'll try to keep that energy high with the sunlight. I am really happy to talk with you all about some clinical trials that we, in the global sense of we, many colleagues around the world are working on towards long-acting antiretroviral therapy with an eye on pharmacoequity. So the first question you may ask is, what is pharmacoequity? It's a fairly new term, we think coined by Dr. Usian in 2021, which, to just read the definition that he coined, is ensuring that all individuals, regardless of race, ethnicity, socioeconomic status, availability of resources, have access to the highest quality medications to manage their health needs. And so I like to start all studies that I do thinking about whether or not we could design those studies and study projects to address access at the same time as efficacy. Actually, in some talks with colleagues who are thinking about bringing things from the lab into humans for the very first time, I have this talk with them right before we even start to even ask, can we think about how this would work in a low resource setting and who could be receiving it and who else we might design this product for before we even know that we can safely put it into humans. So my goal is to make sure that drugs work for more people and more bodies being both, humans as a collective and the diversity that we are. So why is long-acting antiretroviral therapy important in general and important for pharmacoequity? Well, we know that a suppressed HIV viral load is one of the most important things for individual health and for public health, for U equals U. In most countries that are funded or have been supported by PEPFAR, that between 78 and 80% of people who are HIV, who are living with HIV, are suppressed. And among people who are taking antiretroviral therapy, it's really only missing probably 5 or 6% of those people who are prescribed ART who are not suppressed. However, when we look at the US, we're at 66%. And we are just not doing as good of a job as other countries. And there are varieties of reasons for that. I think disparities in healthcare and disparities in the country is one big reason for that. But we like to bring into this topic the inverse care law, which means that the people with the most need are often not served by the optimal care and services. As we know, most products are developed with the low-hanging fruit in mind or the biggest market share. And so bringing this idea of intersectionality and barriers to care to the drug development as we do it is one of the key goals that I think many of us share across industry and public and private sector. So long-acting ART addresses many of the reasons why people are not able to be suppressed, from individual and societal issues like stigma, unstable housing, substance use and mental health challenges, migration, any reason why a person might have trouble putting a pill in their mouth every day or getting that pill into their hand to begin with, or feeling safe having that pill at home. So these are sort of multi-level problems that may impact people differently, but one common thread to address that could be long-acting ART. So there's only one current long-acting ART fully active combination that's FDA approved and being used in some countries around the world. And that's a combination of cabotegravir and rilpivirine, which I'll call cab and ril. It is FDA approved right now only for people with viral suppression. So that's our inverse care law right there. In people who have used this combination in research in the real world, they're very high levels of satisfaction. I can tell you amongst the people that I help take care of, everybody wants access to this option, whether they're having challenges taking to their medication or not, and they would just like to have things be more simple. Barriers right now are that globally it's very difficult to distribute, both in part because the manufacturer is no longer making rilpivirine in much of the global distribution, the need for cold chain, the cost of the drug, medical staff to do all of the injections. It's only given in clinics, not at home. And then, of course, just pharmacy and maintaining all of this. So one of the other really big challenges, both in the US and then increasingly globally, is that the people who are having problems with virologic suppression and virologic failure might have very high rates of rilpivirine resistance because of prior use of Atripla, which was the global first line with efavirenz, or single dose nevirapine for prevention of vertical HIV transmission in years past. So this is not the end-all be-all, even though it's a really fantastic option for many people. So the University of Washington, along with a number of partners, are working on this very issue with a new combination approach. This is called DCNP, or a combination nanoparticle. So unlike a liposome or some of these other kind of versions of conglomerates of drugs with lipids, this is a tightly bound compound that forms a nanoparticle that they call a nano lozenge, just because it's kind of the shape of a cough drop. But the particular one that we were testing in our first in human program contains 3 drugs, tenofovir, lopinavir, and ritonavir. So that's all 3 generic drugs in one nanoparticle, drug combination nanoparticle. And there's an idea and in development a long pathway of several different versions of this drug that have different combinations of generic antiretroviral drugs that can be put into this particle and used as combinations, with the idea that working with global partners like Unitaid and PEPFAR, again someday, that if these medications are shown to be safe and effective, that they could then easily be manufactured globally and distributed in some of the big global distribution programs. So I'll talk a little bit about our first in human study that we just concluded. This is with the TLC ART 101 product. The TLC program is the targeted long-acting combination ART program at the university, which spans from pharmaceutics into our clinical trial space. And so this is at the fixed dose drug. Again, I mentioned lopinavir, ritonavir, and tenofovir. For those of you who are paying attention at home, that's not a current first line treatment combination, but it was developed with this idea that it might be a good drug combination for people who are failing first line therapy, and also for pediatrics. Again, this idea of not leaving people behind. Kids are often the last ones to get access to drugs. So this idea of developing something that would be kid friendly, with many kids around the world having received lopinavir for a couple of decades now. The excipients, or the lipids that hold this particle together, are both FDA approved and used in a variety of other lipid medications. So we had this first in human protocol to give this medication as either a single or 2 injection dose administered near the umbilicus or the belly button, and with the idea that we could dose up or down and decrease or increase the duration of the trial in order to suit what we were finding as this trial unfolded. So this is an adaptive design. And the goal for this was really as a proof of concept for the TLC ART 301 drug, which is long-acting TLD, or tenofovir, lamivudine, and dolutegravir, which is the global first-line generic in much of the world. And the plan for this development program is to partner with, again, global partners like the Medicines Patent Pool and Unitaid to be able to work through both the clinical trials, the manufacturing, the scale up, and the distribution all at the same time so that nobody gets left behind. So these are the results from the first 12 people. That's the entirety of this small trial where we tested this drug combination nanoparticle. You can see on the left at the low, medium, and high doses, the lopinavir lasted for pretty much the duration of the trial for about 2 months. And the ritonavir washed out of the body very quickly. So this is sort of surprising. We're used to having lopinavir have to be boosted by ritonavir. The ritonavir disappears and the lopinavir is there for 2 months. On the other hand, the tenofovir was really gone from the plasma fairly quickly. The half-life was extended up to 4 days in people, which is different from just a few hours when taken as a pill. And then we looked at the intracellular levels of this drug. It lasted for between 7 and 14 days. And then we also did lymph node biopsies that showed that we can get very high levels in the lymphatics of this medication. We were featured in the New York Times. This is one of our research team and one of our participants who offered to literally bare themselves for the New York Times and talk about this first long-acting combination drug that was tested in humans. What we did see, however, were that a number of people had immune reactions to the medication. Everybody on study had a mild and moderate injection site reaction, so that's very common with pretty much everything that we inject into people, and that was not concerning. However, we had a number of participants who had either generalized itching or a rash develop, and we had one participant who developed anaphylaxis or very serious reaction. But through some of the work that we've done, we found that this is not sort of a classical anaphylaxis picture and is something that is probably more likely to be able to be reversed. But it will take a lot of further work to test both the safety of this medication and then also to work on those long-acting properties, since the tenofovir component of it would be what would be moving forward with any next drug. So I want to move on to antiretroviral drugs that are already approved by the FDA and new combinations, but focusing on folks who have unsuppressed HIV. So there's an ongoing study right now on long-acting rilpivirine. This is a study that was originally co-designed by a number of us with the manufacturer, who's ViiV GSK, and is now called the CROWN study, being sponsored by ViiV. It's rolled out in the United States and Spain, and I think opening soon in Portugal, and has enrollment well underway. And the sole purpose of this study is to look and see whether we can expand the use of rilpivirine from people who are virologically suppressed to those who are not suppressed, with a specific focus on people who have had unsuppressed viral loads, difficult adherence, or who've been lost to care. So these are specifically the people who really need this regimen and are considered to be potentially more difficult to reach with care. So this is ongoing. And then there's another drug that is FDA approved, but kind of the lingo that people use is that it doesn't have a dance partner right now. So this is lenacapavir, the scientific advance of the year for PrEP. Previously, it had already been approved recently for treatment in combination with optimized medications in people who've had heavily pretreated HIV. But it doesn't really have, again, this long-acting partner because it's a Q6-month drug. It's the first approved capsid inhibitor with no cross-resistance to other medications, which means it's this perfect drug to test for folks who might have rilpivirine resistance. And so because these are sort of these drugs are already FDA approved on their own, the idea is testing them together, which has already been done in several demonstration projects, but there haven't really been any prospective interventional studies to look at this. So that ACTG is looking at this in 2 different studies. I'm co-vice chair of this study called PALIS, which is a single-arm proof of concept study, which is essentially finding people who have challenges to taking their ART and have rilpivirine resistance and can't take tab and ril, and following them for 2 years and then through a transition of care to standard care to see how well this medication works in folks in this population. And then hopefully soon to follow is the Lancet study, which will be randomizing folks who are failing first-line TLD globally in low and middle income settings, and randomize them to stay on their TLD with enhanced adherence counseling, which is the WHO standard for people who have virologic failure, and possibly switching to a protease-based regimen for those people for whom that is appropriate per the country guidelines. So that's the standard of care versus receiving Len/Cab, again, for up to 2 years. And this will be a large global study. Both of these studies we hope will open soon. But again, the delays in the network funding have really put a hamper on the development of these 2 studies and delayed our plans. So just to close out, I want to talk about how research really brings that pharmacoequity back to long-acting ART. We have the opportunity, especially through federally funded research, to flip back that inverse care law, that the populations who are failed by HIV care are often the ones that are left out of research because they're considered to be challenging to engage in research. But we know how to do that research. We know how to engage those populations and work through community engagement. And so we hope to do that. And also, I really want to emphasize some of the things that, Joe Eron said earlier today, is that public-private partnerships are so important and increase the speed, the resources, and the focus. We cannot go this alone, either as industry or as public-funded folks. We really need to work together. And that research now also predicts the future for HIV care. So any interruptions in our research right now will impact the care to people 5, 10, 15 years from now. But also the current care that exists is necessary to be able to conduct the research. And we really need to be able to have these global partners like UNITAID and PEPFAR and USAID to be able to really do this research and then also land folks who are on research safely back into clinical care, either on the new product or back on the standard of care at the end of the study. This is really the ethical demand. So this is important. And we also really need to move towards testing hybrid clinical effectiveness and implementation studies, where, again, we're looking at how to use the drugs as we are testing to see whether or not they work all at the same time so that we can roll them out more quickly and successfully. So I just want to end with gratitude. This was from our team at Pride and our favorite local supporter, Alexa Manila, who is our favorite drag queen in Seattle. And thank you to all the HIV activists and study participants. And anybody who would like to join our CAB is welcome. We always have hybrid meetings. And so please, whether or not you're living with HIV, click on our QR code and feel free to join us and come talk about research.
Rachel, I had the privilege of talking to you about this talk, and even after watching it for the second time or seeing it, there's a ton to learn and unpack. With the 2 minutes that you have left, I have a question, and I think that it sort of helps hopefully it helps frame the importance of all of this, but also the difficulty of all of this. Can you explain, I'm not a clinical trialist, the enormous amount of effort that I imagine that goes into standing up a clinical trial, and what happens when funding is disrupted, even for 20 minutes?
Yeah, it's hard to encompass that in 2 minutes, but I'll do my best. I can say that as a member of the ACTG network, where our funding has been decreased and where we have had pauses to new activity in our network since March 28th, and we are very slowly beginning to unthaw soon, I hope. What that means is that anybody who we have currently on study at our sites, we are continuing to see them and follow them up, take care of them, make sure they're safe, and collect their samples for later analysis. But it means that we are not enrolling any new participants. We are kind of slowly in the background developing new trials, but we are not getting them out in the field. And we're really not doing any new activity. The clinical trial life cycle is usually about 15 years from the time that somebody has an idea, writing a protocol, filing with the FDA, going all the way up. And so an interruption now is really creating interruptions in, what will become guidelines or what could become the standard of care 5 and 10 years from now, the impacts that we're seeing right now are so much less than we'll see in the future. That said, even a very short interruption is devastating for the fact that we have this huge infrastructure that maintains readiness, just like our public health teams, just like our military. We have huge teams that do all of this, and we can't let them go and then try to hire them back or hire people with anywhere near their skill set and experience back to be able to do this. So there's so much institutional knowledge that would be lost. And so much waste, actually, so much cost when we have to slow down and start again.
You know, I've not heard a lot of people talk about it in terms of readiness, and we think about this all the time when it comes to military readiness or police readiness. This is how we get things done in science too. Rachel,
yeah, I mean,
thank you.
MPOX, right? The way that we were able to do the STOMP study was that the ACTG had this network that was built and ready, and investigators who know how to do this, and therefore were able to stand up an MPOX study in weeks because of this. And if we didn't have that readiness, that wouldn't have happened.
Your work is inspirational. I'm glad you are doing this. Thanks for joining us tonight. We have a wonderful video before we go over to our next speaker. I think. We'll just assume that there's a video.
I'm 68 years old, and I've been living with HIV for 35 years. When I was first diagnosed, there was no treatment for people with HIV, and I had no hope. I learned about and enrolled in an early HIV clinical study. It didn't work for me, but it showed me that there was research into HIV, and I've had hope. In 1995, I started protease inhibitors. Adherence was difficult, and there were bad side effects, but it was a game changer. I stopped going to my friends' funerals and started going to their birthday celebrations. HIV research has given us treatments that make living with HIV manageable, but that's only true for some people. We need to continue to fund and to fight for HIV research. We need treatments that are affordable, available, and accessible for everyone. We need HIV research. We need an HIV cure. Thank you.
All right, we are ready to move to Carlos Rodriguez-Diaz, who is a champion of the people and whose work is absolutely inspirational. I'll pass it over to you, Carlos.
Thank you, Josh, and good morning, good evening, buenos días, buenas tardes, bonne nuit, buenas noche. It is my pleasure to be here today, and I'm very honored to be part of this initiative. This is very dear to my heart as a scientist, but also as a community member, and as a person who's been navigating the world of HIV. I was born in 1981, and I don't share that for you to do the numbers of how old I am, but just to share that I was born in a world with HIV. I don't know a world without HIV. And that was part of the inspiration for me to engage in HIV research, but most importantly, to engage in HIV research that was meaningful for the people that were disproportionately affected by HIV. So I'm honored to spend the next few minutes sharing with you my experience in conducting community engaged research, mostly to move the science so we can address the needs of the communities. You know, I have learned with community partners that we've done a really good job in identifying what are the problems in the HIV epidemic, but we haven't been that great in actually addressing those issues. We have developed interventions, but then the communities that could benefit from the interventions have to wait years to benefit from those interventions. So today, I will share with you my experience of conducting research with communities that have helped us scientists in making sure that the interventions reach out to the populations that really need them. I also want to share a little bit about me because it's influencing what I do and the way I do the work that I do. I am Puerto Rican, very proud of having grown as a scientist in Puerto Rico and conducting HIV research, and also now as part of a larger community of scientists in Puerto Rico, in the Caribbean, but also in the continental US. I feel that I belong to a community, but it's the community of people that have been disproportionately affected by HIV that have guided me to do the research that I feel is more relevant to the community. I also want to recognize that sometimes we see science and research as a field that people that are affected by or that comes from the communities that are disproportionately affected might not have a place. And the scientists are other people in academia or in research institutes, but we belong to research, either as a scientist or people that can inform the work of scientists. And I hope that my participation here also serve as an opportunity to some of you that are watching now or seeing the recordings to find a space and see that it's possible to contribute to ending the HIV epidemic and to make a difference by doing research that is meaningful to our communities. So I have a couple of slides that I will share with you. And, you know, one of the challenges that we have in HIV is that we have done a really good job in identifying problems and also coming up with solutions. But we know that there is a gap between identifying solutions and interventions that can address the epidemic and actually having the communities using those interventions so we can reduce either negative HIV care outcomes or reducing new infections. The literature have shown that it could take almost 17 years from designing studies that help us coming up with interventions to having the communities that could benefit from those interventions using that interventions. Which is worse, is that it could take more than it's less than 40% of those interventions that we research on that actually get into the community and have an impact in the services, either through policies or direct programming serving the communities. That's problematic. And through the years, we have scientists come up with a field of work that we call implementation science that basically is providing us with the guidance, the frameworks to study the methods that we are using to scale up interventions that we know are working or could work in addressing issues affecting people that are more vulnerable to HIV. So implementation science is becoming more and more visible as a way of moving from doing research, but also addressing the needs of the community. And why is this important? Well, because if we use as an example the National Institute of Health in the United States, the recently appointed director of NIH have released a statement on advancing the NIH mission through unified strategy. And one of the core components of that strategy is implementation science. It's advancing the science of HIV research into the practice. So therefore, there seems to be an interest in moving to actually using the scientific knowledge that we have with communities and the people affected with HIV so we can achieve the goals of ending the HIV epidemic. And as our dear Tony Fauci have shared with us, we can end the HIV epidemic domestically in the United States, but also globally. After 44 years of research on HIV, we have evidence of all these factors that we can intervene to prevent HIV that now comes from testing to biomedical interventions to counseling, but also to treat HIV. That includes people knowing their status, linking them to care, providing successful treatment, and advancing to the point of achieving viral suppression so we can have a community advantage for individuals who can achieve viral suppression, and with that, reducing the likelihood of infection. So we can end the epidemic. We are able to know all this because we have done a lot of research that allow us to identify interventions that help us identifying strategies for HIV testing, engaging people in care, providing PrEP care or counseling for couples. We have multiple interventions that we are aware of that can be implemented. But how is that we know that? Well, we know that because of the communities. I argue, and I hope that some of you either agree with me or will understand better after this few-minute presentation, that communities started the end of the HIV epidemic. We have coined the term ending the HIV epidemic over the last 5 years, but the truth is that since the beginning of the epidemic, we have envisioned a world without HIV. And the community have had a core role on doing so. We haven't reached that point where the community is fundamental in responding to the epidemic, but we are moving to that point. And over time, we have improved in our way of engaging communities so we can conduct research that is meaningful, that is representative, and that can be translated successfully. In this, I will show a couple of pictures, including some articles and some of the community leaders that inspire my work. In these slides, you can see the title of an article from Marcia Ellis. She used to be the director of the community engagement component at the DC CFAR. And by the way, I mentioned the CFARs as a component within the NIH use of resources that have expanded the ability for several jurisdictions to support meaningful and community engaged research. And in the second picture, I'm glad to share an image of Rosa. Rosa is a Puerto Rican leader within the HIV community that gave us many lessons of how can be more inclusive and responsive to the needs of the population affected by HIV. And the engagement of the communities in the response to HIV is not new. It's been happening since very early in the response of the epidemic. Here I'm sharing an image from ACT UP, which was fundamental in the response to the epidemic in the 80s and 90s. They made demonstrations that changed the world, that changed the way we do research, not only HIV research, but public health research. Nowadays, we are kind of accustomed to have community engagement in our research activities. But the truth is that it was not like that just a few years ago. But because ACT UP actions and the leadership of members of the communities affected by HIV, now we have a model or several models that are inclusive of the community. And in fact, we have had funding opportunities that require the engagement of the community. That is not happening in a vacuum. It's happening because activists and community leaders engage in visibility, in pushing scientists and pushing authorities to be visible and to make sure that their voices were included when prioritizing and deciding what kind of research, where we were researching, and for what. ACT UP was all over in the United States and in other spaces. Here is a picture of ACT UP in Puerto Rico. ACT UP was fundamental in including Puerto Rico in clinical trials and increasing the diversity of people that were participating in clinical trials back in the late 80s, early 90s. When we have ACT as part of clinical trials, we because of ACT UP, we were able to engage people in clinical trials that otherwise wouldn't have any expectation in terms of care because there was nothing but to be part of clinical trials. So that's how the community disrupted these systems in a way that we could do research in a different and more meaningful way. And if we look to more recent history, the fact that we know that PrEP works, that we can have PrEP on demand, and now that we can have a twice-a-year injectable, is because we have members of the community volunteering to participate in research enterprises, not even knowing if they're getting a good treatment or if they are a control group, but because they care and there is an interest in contributing to their communities because they know the impact that HIV is having. So we have all this scientific knowledge because of community engagement. And that community engagement on HIV has helped us addressing other epidemics, outbreaks, and pandemics. I've been fortunate to be surrounded by very good mentors and other collaborators that have helped us translate what we have learned from the HIV epidemic to all these other public health emergencies. Just an hour or so ago, we were talking about Mpox. We were able to respond to the Mpox outbreak because we've learned of how the community can support the HIV research and HIV response. And we also learned from the HIV community and community engagement on how to respond to the COVID pandemic. We unfortunately knew very early in 2020, when the pandemic was at the heights in the United States and globally, that we were very likely to see the disparities that we know are affecting people with HIV, affecting communities of color and those that were disproportionately affected by other social determinants of health. I want to share with you some of the work that we have conducted, funded by NIH and other federal institutions, that is helping us to move forward on how we can work with communities to do better science. Recently, we were able to publish a paper that we titled "Community Start at the End of the HIV Epidemic." And the beauty of this paper is a combination of scientists and implementers, people working in the field, helping us identifying how is that we can work together. And as you can see on this table, and I'm sorry that it perhaps is too small, but the paper is of open access, so you can look for it. There are some areas of work that we clearly detail as implementers and scientists on how we can make decisions together so we can be successful in conducting research that is meaningful for the community. And as you can see, this goes from establishing goals that are important for the community and all the way to sustaining research and the outcomes of the research that we do. We have also done work on language justice. Working with Latinos in the continental US, something that we have learned is that language matters. And it's not only a matter of English and Spanish, it's how we do the work with the Spanish-speaking populations and those that have learned English as a second language. And we have published, again, working with community partners, what are the takeaways of doing research with bilingual or multilingual populations? And the takeaway that you can take a closer look in the publications is that it is important that the community is involved, from conceptualizing the research all the way to dissemination of the research findings. What we documented is that the community engagement help us to getting better in creating documents and instruments that were better in capturing what we were learning from the community, so the research instruments were better. And when we were intervening, the intervention materials were also responsive to the community. And within the Latino community, I believe that one of the main challenges that we have in the continental US is that we think that all Latinos are equal. Well, we are not. And most recently, we have been publishing on dismantling that idea that we are all the same. It doesn't mean that we are that different to the point that we cannot do research that is comprehensive of the Latino community, but that is important to understand that there are differences. And by engaging the community, we are better in responding to those changes or those differences. So in a nutshell, and closing up my presentation, I want to share some of the lessons that we have learned. First of all, community members have a role in research, and the role is from the beginning. And it's even better if the community can own the research and the research question comes from the community. And if you're a scientist, you might say, well, how that happens? Well, talk to the community, spend time with the community. Yes, it takes longer. Perhaps you have to spend time outside of your office, and you have to spend time in wherever the community is, wherever the people affected by HIV are. Well, then that's good. If it takes longer, it's fine, but it's more meaningful. If you are taking the advantage of learning from the community, hire them, pay them. You can hire people from the community that can be extremely resourceful in facilitating engagement, access, but also bringing that knowledge into the work that you do, and nurture those experiences. It takes time, but also over time, it becomes more and more rich. It's lovely. And as a closing, I have to plug again on the work that we do in Puerto Rico. Thanks to the funding that we have received from NIH, we have a model now that we have implemented for 6 years, in which government, community, and academia sit down every 3 months, and we talk about what's going on with the epidemic, and we have come out with ideas for research services, and we share our experiences in a way that built a strong response to the epidemic locally. And with that, I will stop. I think right on time. Thank you for the opportunity, and hopefully we'll have some time for questions. Thank you, Josh.
You are perfect on time, and a master at helping us understand how to do and start the wheels of community engaged research. And that was actually going to be my question that you stole from me at the end, but maybe I'll just ask anyways. There are presumably young and seasoned researchers on right now. In a nutshell, what is your one piece of advice of getting started in taking a more community engaged approach?
Talk to people. Don't think that you know everything. You know what you think is the science, but the day-to-day experiences, the activities that happen in their communities, we are not aware. We are not them. We need to close that gap between me and them so we can be us, and we have to invest in that process. And depending with which community we're working, it takes different forms. Yeah. But just spend time, listen. You're not the expert of the lived experience of any community.
I feel like that's a mic drop right there. Thank you. I'm going to kick you off screen and go to our next. Carlos, a total pleasure. Thank you. And to close out this hour, we have Kara Chu, who's a physician over on the West Coast in California at UCLA. And I will hand the mic over to her to talk about a topic that we've sort of touched on through the day. And I'm excited to hear what you have to say on this.
Great. Thanks so much, Josh. I'm really excited to be a part of this great event today and to talk with you about hepatitis C, which disproportionately affects people living with HIV. And in the next 13 minutes, I'm going to review not my own research, but the research of many others that has really been critical to understanding every aspect of hepatitis C infection, what it is, how common it is, how it spreads, its impact on health, and also how research has been really fundamental to discovering curative therapy for hepatitis C, and how it's going to be really fundamental to achieving elimination of hepatitis C across the world. And just because today is focused on HIV research, I will emphasize some of what we've learned with respect to hepatitis C for people living with HIV. And just starting with that very first topic, which is just identifying hepatitis C, I just think it's fascinating. It was an unknown virus that was causing liver disease associated with blood transfusions in the 1970s. And they didn't have a name for it and had identified hepatitis A and B, and it wasn't those. So it was first called non-A, non-B. But then it was identified through research in 1989. And then shortly thereafter, blood tests were developed to be able to diagnose people with hepatitis C. So really through it all, research has been essential to diagnosing and identifying hepatitis C to today. When we think about the burden of hepatitis C globally, there is a significant burden. 2022 WHO estimates put the prevalence of hepatitis C at about 50 million living with hepatitis C, and that's with detectable virus in the blood worldwide. And in 2022, there were approximately 1 million new hepatitis C infections. And people living with HIV have a higher burden of hepatitis C, mostly through shared routes of transmission. And it's been estimated over the years that maybe up to 2 million people perhaps are living with both HIV and hepatitis C, and upwards of a quarter of people with HIV have had hepatitis C at some point. Today, the estimates, at least in the US, look more like about 5 to 10% of people who have HIV are also living with hepatitis C chronically. But there really aren't great estimates in many parts of the world. Hepatitis C is an infection that primarily affects the liver. And for many people, they don't have any symptoms when they acquire hepatitis C or for several decades living with hepatitis C. But over that time, liver disease can progress and result in cirrhosis, liver cancer, and death in some, and perhaps a need for a liver transplant. And so while there aren't any symptoms that people can always identify, research has demonstrated how very common symptoms are actually that aren't really obviously directly from liver disease. Symptoms like, or conditions such as depression, feeling really tired, having trouble thinking, all of these symptoms that affect quality of life. And hepatitis C, which is a chronic infection with chronic inflammation, just like HIV, is also associated with other conditions outside of the liver. Heart disease, autoimmune disease, kidney disease, cancers like lymphoma, diabetes, and insulin resistance. So it's a systemic disease, really, not just a liver disease. We've learned a lot through research, too, and surveillance about how hepatitis C is transmitted or passed on. And the main way is through direct blood exposures, particularly injection drug use, which accounts for about 60% of new infections in the US. We've also seen that it can be passed on through shared devices for non-injection drug use. It can be passed on mother to infant. And then particularly among men who have sex with men and men who have sex with men who also have HIV, transmission through sexual activity has been more recently identified in the last, I'd say, 10 to 15 years through research. And when we look at the rates of new hepatitis C infections over time, and this is CDC data, for a while we were seeing that these were really climbing for some years in all age groups, across sex, and race and ethnicity groups, but especially among younger people, 20 to 39 years of age in the US, with this epidemic driven especially by the opioid epidemic and injection drug use. Now you can see that in the more recent years, it looks like the rates of new infections have actually been leveling off or perhaps decreasing a little bit in many groups. I do wonder how much COVID had an impact on these measures and actually detecting hepatitis C. So research has also, though, taught us where we're really falling down in preventing deaths and where there are racial and ethnic disparities in deaths due to hepatitis C. In this paper, also using CDC data, hepatitis C-related mortality rates differed by race and ethnic group in the US and were highest among the American Indian, Alaska Native, and Black race and ethnicity groups. And specifically for HIV, people living with HIV and taking antiretroviral therapy, having hepatitis C increases mortality. And it's associated in this study with a 50% higher risk of death compared to people without hepatitis C. And in this study, 20% of those deaths were liver-related. And we've learned from research that liver disease is accelerated. It progresses faster in people who also have HIV, with 3 times a greater risk of progression to cirrhosis or decompensated liver disease. And that would be conditions such as ascites or bleeding from dilated blood vessels. Another thing we've learned from research is that people with both HIV and hepatitis C have more inflammation and immune activation than people with HIV alone, which contributes to many of the systemic complications that I mentioned earlier. But the good news is that we have something that works. We have hepatitis C treatment that's very effective. And we saw this before with people with hepatitis C alone, but there are great data that hepatitis C treatment reduces deaths from all causes in people with HIV. And so in these figures, what we're looking at are these lines. And if the line is going down, that reflects people dying during follow-up in the study. And if it stays flatter, going across, that means people are not dying. And what we see in each of these boxes, overall mortality, liver-related deaths, non-liver-related deaths, non-liver, non-AIDS-related deaths, is that the line is flatter and many fewer people are dying if they have sustained virologic response, or SVR, or what we consider cure after hepatitis C treatment, than if they aren't cured with treatment. And so that's great news. There has been a lot of progress in treatment options for hepatitis C over the last 30 years now. And what's given in this figure are hepatitis C cure rates, those SVR rates for people with hepatitis C alone in the blue bars, and then for people with hepatitis C and HIV in the reddish bars, and that's over time. And you can see early on, we really just had data for people who had hepatitis C alone, and that's often common in studies and clinical trials, which is that drugs or interventions are tested first in people without HIV, and then later in people with HIV. And so early on, we saw very poor response rates with interferon therapy, which is an injectable chemotherapy-like drug, given for a very long time, almost a year. Only 20% cured. And so the shorter the bars, the worse. And we want higher bars. And then with the addition of ribavirin, then those rates improved a bit, but still not great, 40%. And then with that same therapy, people with HIV did worse, didn't have as great response rates as people who had hepatitis C alone without HIV. But then there was a lot of drug development. Treatment response rates really improved. And they started to match up more in people with HIV or without HIV as far as the hep C cure rates. And so that's great. And what we have here are some studies of clinical trials that were specifically done of these all oral, highly effective hep C treatment regimens called direct acting antivirals, studies done in people with HIV. And we're really today at a place of 98% plus cure with these all oral therapies, which are taken for 8 to 12 weeks. And one of the biggest discoveries that changed hepatitis C treatment and had a huge impact on human health was the discovery of the drug sofosbuvir. And I just found this article really interesting. And I will basically quote them, which is that many of the most transformative drugs from the last few decades that have had a really groundbreaking effect on patient care were discovered and developed in part based on funding from the NIH and other public sources. And this was funding to academic medical centers where a lot of early drug discovery happens, to government laboratories, and then to startup companies. And in the case of sofosbuvir, which was approved in 2013, it was the first in its class of antiviral drugs, highly effective, really safe, well tolerated. The NIH investment, both in direct funding for this work and indirect funding, some of which went to HIV drug development, that investment was estimated in this paper at about $60.9 million US. And that what an incredible investment that was for this life-changing drug. So we've got great therapies. Why can't we cure more people and eliminate hepatitis C? Because we really haven't gotten that far. To me, and this is my perspective, this is an implementation problem. So here we have all the steps in the hep C care cascade, from diagnosis to confirming infection and need for treatment, to linkage to care, to getting actually started on treatment and completing treatment, and then finally achieving cure. And we know that there are barriers at every step in this cascade, and particularly worse in people with HIV. And I've listed on the right-hand side a number of the barriers that have been identified, particularly in cohort studies or case series for people with HIV, barriers or characteristics associated with not initiating treatment. And some of them are no longer relevant in the sense that the drugs we have today don't cause the same side effects and toxicities that make us worried about treating with them. And others, they still persist, but really they shouldn't, right? And so these are barriers that we really need to address. We've learned through research how to more efficiently deliver hepatitis C treatment and try to address some of those barriers. And this is one example. This is an ACTG study, an NIH-funded study through the ACTG network called MINMON. And here they really tried to simplify testing for Hep C, liver disease stage, and they provided the whole treatment course up front. Required no in-person visits after the first visit. And what they found was great success with high cure rates among people with HIV and hepatitis C or hepatitis C alone. But we have a long ways to go. This publication just shows an example of how well we're doing with getting people cured or cleared of hepatitis C using lab data out of Quest. It's not great. What we want to see more is that some of these match up better. So here in this study, in this graph, initial infection really refers to people who actually had detected virus in the blood of Hep C, so they really had an indication for treatment. But the cured or cleared rate is much lower than this bar. So really what we want is to see that they match up, that everyone is diagnosed who has it, and that everyone is treated who has it. So globally, how are we doing with Hep C elimination? This map shows how different regions of the world are doing in terms of achieving programmatic targets. Red is not on track. That's a lot of the parts of the world. Yellow is working towards making some progress, and green is on track, and that's few places. So what about the future? I've talked a lot about all our successes, but the next horizon and really achieving hepatitis C elimination, I think, requires a lot of research funding, both for more rapid and better diagnostics, point-of-care tests to identify who needs treatment, long-acting injectable Hep C treatment. Imagine a single shot instead of 8 or 12 weeks still of pills that could cure someone of hepatitis C, and then hepatitis C vaccine. I mean, that's really essential to prevention, but there are a lot of challenges to developing such a vaccine. But research will be essential to try to approach all of these. And then finally, for what we have right now, highly effective drugs, we need implementation strategies that are really effective, and we need research funding to do that. So I think that's all I have. Just wanted to acknowledge a couple sources, Dr. Wiles in Colorado, and I'll stop there.
Kara, first of all, that was like the tour de force of hepatitis C. You made it so accessible, why it's important. I'll admit hepatitis C is tied with my first loves in medicine. I have to say that. And I also have to say that because Dave Wiles is my boss. So I'll say it out loud.
It was my first love too.
You bring up so many things that I wish we had more time to talk about. There's a question in the chat, and I'll sort of expand on it, but it says, how do we continue to learn lessons from one virus to eliminate another? And as you've sort of pointed out, we haven't eliminated it either, that we're talking about right now. As you approach, you mentioned implementation, but as you approach these two viruses, both together and separately, how do you think about elimination and what sort of some of the missing components are right now?
Yeah, I mean, I think that there are so many parallels, right, in terms of at every step of the care cascade, and at diagnosis, linkage to care, access to treatment, getting broad coverage of treatment. I think that there's so many parallels, and what we learn in terms of what's successful in one setting could possibly inform success in another. Also, co-locating treatment of both, because there is a higher frequency of having both HIV and hepatitis C. I think trying to tackle more than one problem at once is really essential. Yeah. I also think it's really we learn a lot. I mentioned that some of how Hep C drug targets were identified and drugs developed was from HIV funding. And there are a lot of similarities in terms of these drugs. Like emtricitabine works very similarly to sofosbuvir. And then in terms of a vaccine, we have a lot of the same struggles in terms of the diversity of hepatitis C, diversity of HIV, understanding immune correlates of immune control, what we need to generate in a vaccine response to prevent or control HIV. There's so much overlap and so much that the field can learn from each other.
And I think just to close out, I do think also there's a lot to be said about I mean, 10 years, 15, I should say 15 years ago, this was not what we were experiencing.
Yeah.
And it sort of is a story of what is possible when we invest in research, when we invest in humans, when we set our minds to something. I think that Hep C and HIV have this parallel also of when we actually put our minds to things. No one 44 years ago, 45 years ago could have ever imagined where we would be now with HIV. And honestly, 15 years ago, nobody could have imagined that we would be talking about one pill once a day and getting rid of Hep C. So I really appreciate you bringing this to the forefront and talking about this tonight, spending 20 minutes. We're going to pass the baton to others, myself not included. So Kara, thank you.
Thank you.
We'll bring on for the next hour, cue music.